we have a very full waiting room on our webcast. And so as people get settled here in person, we have a number of people that are already up in the Zoom. So thank you everyone for joining us this morning, and I'm glad to see so many people both in person and present in our webcast. We're delighted to be able to see you all virtually and presently and give you an update on the R&D progress that has been made in plant health. And of course, this is the first event of a series of two. We will have a R&D showcase as well, covering our human health programs on Thursday. And so what we're going to do today is we're going to give you a rapid fire of all of the novel data that has been generated really in the last 6 to 9 months. And so the way in which we're going to do this is I'll give you a brief overview of our global progress. We'll turn it over to Mark to give us an update on our plant health strategy. Mark, who's our Chief Commercial Officer and General Manager of our plant health business. And we'll turn it over to a number of our scientists who are going to be talking specifically about some of the data that we'll be sharing with you guys today. I'm going to ask you to hold questions till the end. We have about 150,000 slides that we want to get through over the next hour and a half, and then we'll try to leave time at the end for questions. So again, delighted to have you all here. What we want to give you is an overview of how GreenLight is responding to the need to develop new actives for food protection, for food security globally in a much faster period than what we have been able to do historically, and to do so while preserving the biodiversity and the sustainability of our food supply. Obviously, GreenLight is a public company, and we're going to be talking about things that will occur in the future if everything goes according to plan, and therefore I need you all to make any investment decisions that you may make based on your own diligence, and to obviously consider the fact that we're going to be projecting things that will occur in the future. So let me jump right in. The way in which we grow food is not good enough. We are 8 billion people on the planet. We'll be 10 billion people in the next 10 or 15 years. Our population is growing fast, and it's demanding higher quality foods. At the same time, we have to be able to feed this 25 or 30% more people with about 30% less land than what we have historically done. In order to mitigate the rapidly warming planet situation that we actually have to restore rainforests and jungles and permanent plant crops that retain carbon underground, as opposed to taking land further away from those permanent carbon deposits to make room for agriculture. That is a tall order, because what we're seeing in the background is rising resistance, demanding higher quality protein. We're seeing changes in regulation and consumer behavior that demand that we grow food in a more sustainable way that is more respectful of the biodiversity of the planet. We see major changes coming to the way in which we grow food globally. We see precision agriculture not being a nice to have, but truly a must have. We see the rise of automation and the rise of artificial intelligence applied to how we grow food. And we see that none of the traditional technologies that we have relied upon for the last 100 years, mainly chemicals, can react in the time that we need them to react to the growing needs of farmers and the consumers around the world. What farmers are facing is unprecedented. The 3 big R's. Resistance. We're seeing resistance from pests across the board, insects, fungi, weeds, developing against the chemical tools that farmers have historically had, now growing exponentially. Fungal resistance, which used to grow linearly over the last decade, has in fact grown exponentially, and that is a direct result of global climate change. We see that trend accelerating, not slowing down. We also see consumers demanding that no residues, no chemical residues be found on food, especially residues that are known to be harmful to human health or to biodiversity. Whether real or perceived, the threat of chemicals in food is something that consumers want to avoid. We need solutions that leave no trace in our food. You all know that I started this company because my son Alex is highly sensitive to organophosphates. Organophosphates are still used as insecticides in about 50% of the crops globally. That is unacceptable, and that needs to change rapidly. And then finally, regulation. With consumer changes, with the need to preserve biodiversity, to preserve soil fertility, governments are responding by tightening the regulation against chemical pesticides. It is becoming increasingly more difficult and more expensive to develop new chemical tools. And that is reflected clearly in the data that we see. Over the last few decades, new chemical actives developed by the major agricultural companies have been reduced by over 70%. The pipelines of those major agricultural companies are drying up, and the cost of developing new actives keeps on increasing. At the same time, farmers have less tools to treat emerging pests and pests developing resistance. If we just project those trends forward, within the next 20 years, unless something drastic changes, we're going to have a global food crisis that is going to be unprecedented. So the opportunity has never been better. The opportunity has never been clearer. The task has never been more concrete than for new companies, innovative companies, to develop solutions that can answer the call, that can bring active ingredients that are capable of overcoming resistance, that are friendly to pollinators, to soil microbes, to biodiversity, and that are acceptable to consumers and regulators. The time has never been better for a company like GreenLight to bring its products to market. And we are responding to that call. We went public almost a year ago now, more than a year ago now. And in the last 12 months, the progress that has been made has been nothing short of spectacular. When we went public as a $1.5 billion company, we had one product under consideration by the EPA. Today we have two, with one of them on the brink of being approved. When we went public, we had one mode of action demonstrated. We had shown in large-scale field trials that we could kill Coleoptera. And when we showed people our data, people said, so what? We all know that you can kill Coleoptera with double-stranded RNA, but we believe that's the only thing you can kill with double-stranded RNA. Today we have demonstrated that we can stop Coleoptera, that we can stop Lepidoptera, that we can stop fungal pathogens, that we can now begin to stop weeds. By the way, Lepidoptera, fungi, weeds, which account for the majority of the destruction of our crops globally, we were told it was impossible to control them using RNA. And of course, here at GreenLight, we're in the business of doing what others think is impossible. We had also only demonstrated one mode of application. We had only demonstrated that you could spray RNA on crops and have a foliar applied, in that case, an insecticide. Today, we have demonstrated that we can continue to apply foliarly for a broad number of crops, but we can also apply through the root, through coating of the seed, and more recently, by spraying an RNA that becomes systemic to the plant, opening up an entire new category of products that we can bring to market. And finally, when we did our de-SPAC, again at a $1.5 billion market cap, we had enormous regulatory risk ahead of us. What we believe today is that we have mitigated that risk in a very significant way, and we will talk about it more in detail later on. The next two are what I call the money shots. These are the slides that you have all been waiting for. The first one shows that we are able to control fungal pathogens on a variety of different crops, as well as, and in some cases better, than the chemical actives that are out there. This is a picture on the left. You have a strawberry coated with Botrytis cinerea, gray mold, and on the right you have one that has been treated with our double-stranded RNA. In the middle, you have a cabbage plant that is affected by diamondback moth. On the left, you have the untreated version. On the right, you have the version treated by our double-stranded RNA. And then finally, you have soybean roots that are affected by a fungal pathogen whose name I am not going to be able to pronounce, and this is delivered through the seed, and through the seed we're able to arrest the development of that fungi and rescue the plant. And one of the most exciting things that we have seen, and this is a preliminary result, but nevertheless extraordinarily powerful, is we can spray an RNA into a weed. This is horseweed, which is a massive issue in soybean crops around the world. And what we have shown is that we can arrest the growth of that weed, opening up potentially the entire herbicide world to double-stranded RNA and GreenLight. We have also worked extremely closely with the EPA to mitigate the regulatory risk for this new technology. we initially showed the EPA all of our manufacturing data, and we were able to show them that we can produce an RNA species that was very well chemically defined, that corresponded to a new mode of action. That was a twofer. Not only were we bringing a completely new molecule to the regulatory agency, we were also bringing a completely new way in which we produce that molecule. And it took a while for the EPA to get through our first submission. But we selected the EPA because of their commitment to embrace these novel technologies that have the potential of providing powerful tools for farmers while respecting biodiversity, while promoting environmental health. And we actively engaged with the EPA throughout this process. We found in the EPA a willing partner, a rigorous partner, that allowed us to demonstrate the safety and efficacy of our products. In the process, we both learned, the EPA learned about RNA and we learned about the regulatory process. And we had to communicate constantly about how RNA could be used gainfully in farms across our country. The regulatory learnings that we gained through the Colorado potato beetle submission are now being applied across our portfolio. We are predicting what the EPA is going to ask us for and doing those experiments and gathering that data ahead of time. And then finally, we expect the EPA to give us experimental use permit for our Calantha product shortly, and we expect to gain regulatory approval to commercialize Calantha this year. If that happens, that would constitute the first ever sprayable RNA ever approved through a completely new synthesis process. So in a way, it's not a surprise that it took a little bit longer than anybody anticipated to get this product, but what we believe is that going forward, the rest of our portfolio will be accelerated. It is the era of RNA. We are coming to a point where the maturity of the solutions that we have previously had is coming to an end. Bringing a new chemical to market is more expensive and more lengthy than ever. And that is a result of our need, of our society's need to preserve our environmental health. Not just because we like our planet, but because otherwise we won't be able to grow food in the future. Because we won't be able to feed the soon-to-be 10 billion people living in this planet. Because we won't have the ability to continue to grow food and feed everyone. As we went from natural products that were naturally protected of crops to basically medicinal chemistry, we were screening hundreds, if not thousands, of chemicals against pests, to what we have today, which is rationally designed, highly effective and specific RNA targets. What we have been able to do in the process is to significantly reduce the time and the cost of bringing new active ingredients to the market. When Calantha gets approved this year, again subject to EPA approval, it will have been about 5 years from the day in which we first synthesized Calantha. That is less than half the time of what it takes for a new chemical to be registered. It is also about 1/4 of the cost. Today, developing a new chemical solution can take as many as 11 years and rising rapidly. This was only 8 years a decade ago. So this process of approving new chemicals is not going to get any shorter, it's in fact going to get longer. And it can cost up to $500 million to develop a new chemical solution. The reason why we can do this is because we have developed a platform, which we call the Green Box, that enables us to rapidly identify triggers in the target pest. That is, genes that are susceptible to our double-stranded RNA that can be gainfully used to stop that particular pest. And at the same time, through our bioinformatics engine, are going to be safe for non-target organisms. Non-target organisms, of course, include pollinators, other beneficial insects, humans, our children, and other parts of our biodiversity that we rely upon for sustainable food production. We can then iterate, and we are the only company in the world, the only RNA company in the world who can do this, where we can synthesize hundreds of distinct RNA species that we can screen in the greenhouse, that we can screen in the field, that we can screen in the lab, and very rapidly come up with solutions that are going to be appropriate for the farmer needs. By the way, we can also do this for human health applications. And so I'm just giving you a preview while the train goes by of what's going to happen here on Thursday. Now, what does it mean to have a platform? A platform is only as good as the products that it generates. So what I can tell you is this is a very powerful platform that has demonstrated its capability. This is not theoretical. This is a fact. And the fact is measured by data. GreenLight today has 7 novel actives, new actives with new targets, with new modes of action in field testing today that we expect, again, subject to regulatory approval, to be in the market by 2026. 7 novel active molecules. If you don't think 7 is a lot, just consider that Syngenta and Bayer, the 2 largest agrochemical companies in the world, have 6 and 4 respectively. Think about the power of this platform, that with a budget that is less than a tenth of the budget of the major agrochemical companies has, in a matter of 4 years, generated more targets than the armies of highly sophisticated researchers working at those major leading companies. It is the power of our platform that allows us to rapidly identify targets that are going to be efficacious and are going to be safe. And our synthesis platform that allows us to make those targets rapidly, screen them, scale them, and bring them to field testing. We are incredibly excited about the opportunity that GreenLight is in front of. These 7 products have the potential of bringing revenue in a market that we measure at about $6 billion. These 7 products address some of the world's largest needs, address fungal pathogens that are increasingly develop resistance to the existing chemical fungicides, address insect targets that have shown to be multi-resistant to insecticides in crops that corresponds to hundreds of billions of dollars of crop value. We are very excited about the opportunity that we have to shepherd and steward this technology and bring it into the market. And as you know, the elephant in the room is that our stock price has taken a little bit of a hit since we went public last year, and we obviously will need to raise capital to bring all these products to market. And so we're taking every measure that we can to ensure the sustainability of our company, that ensure our continued access to capital, that ensure that we're being as effective and responsible with the capital that we do have. So as you have seen, and you will see in the presentation today, we have prioritized the value creation milestones, meaning prioritizing the commercialization of Calantha and our pollinator protector product that Mark will talk about. We have also identified, as I showed before, that we can now target pests that were believed to be outside the reach of RNA, fungal pathogens, seed delivery, weeds potentially, and those are of broad interest to the agrochemical industry. And so we are in active conversations with potential partners around the world to be able to ensure that we develop those products in the most capital efficient manner. We're obviously engaged constantly with the market, with investors and potential partners to figure out the most capital efficient way to bring in these solutions forward. And finally, we're actively considering every alternative that we have to be able to extend our runway. This is an exciting time for GreenLight. For the first time in history, RNA is on the brink of being approved as a sprayable product. For the first time in history, we shattered the preconceived notion that RNA was only going to be ever gainful to control Coleoptera. We have demonstrated that we can have control that is comparable to the most accepted chemical standards for fungal pathogens that routinely cause tens of billions of dollars of damage to our crops. And we have shown that we don't have any of the unintended consequences that are common with chemical solutions. If we believe that it is our responsibility to find solutions to some of the world's biggest problems, like we do at GreenLight, then this is one of the most exciting companies that we have the honor of working for. So thank you all for coming and seeing us this morning. I hope that you will find this morning productive. We're gonna nerd out on you guys. We're gonna go right into the data. We're gonna show you data that is brand new, that is exciting, that is truly groundbreaking. So thank you again. Hold your questions till the end, and I will turn it over to Mark.
Okay, thank you, Andre. Good morning, everybody in the room and everybody online. I'm going to give you a quick glimpse into how we are thinking about the GreenLight Plant Health business. That is, what is our plant health strategy? Well, you know, as the title of this slide says, we're working to be a leading developer and manufacturer of RNA. Our base technology is the ability to manufacture RNA that is great for agriculture. But you know what? There weren't any products to manufacture that for. So that meant we had to get into the product development business, the product discovery, development, registration, and commercialization business. There was no choice because there was no other market for the RNA that we could make at that point. So we're an RNA manufacturing company, but one with a mission, and that is to create a whole new line of products based off RNA. And so with that ability to create and that need to create, the need that Andre just described, comes a great responsibility. We have to do it properly. And so, when you think about the strengths of RNA, the fact that it is highly selective, you design it to do something and it does just that. That means that we leave everything else alone in the environment. Our footprint is very small. Our ability to help sustain our farming partners, help sustain the biodiversity within the agricultural community is tremendous. And we must make sure that we keep that in mind as we develop our products. We have to build a portfolio, and I'm going to spend a little bit more time about what that portfolio is, how we think about it, and how we derived it. But effectively, it is designed to maximize our early chances of success and continually challenge us to access bigger and bigger markets as we go through. And finally, as the creators of this new world, we need to look after the technology. Stewardship comes in many forms. We need to make sure that we're making the right recommendations for safe use in the environment, that we protect, we avoid the issues of registration, that we design the products to have no off-target effects. Okay, it's one thing to say RNA is good, it has no off-target effects, but you need to know that. You need to demonstrate it. Regulators need to understand it. And we see it as our mission to make sure that we enable that to happen. We want everybody to believe what we believe, that RNA is effective, it's potent, and it's safe. And that is part of our mission to be great stewards for this technology. So, I talk about building a portfolio, leveraging the skills that we have, developing those skills and the know-how we have, and as we work through that process to expand the market. So how do we do that? Well, what we did was we sat down and we thought about, well, the first thing we need to do is to prove. We need to prove that it works. We need to prove that we can make it. We need to prove that we can register it. We need to prove that people will buy it. And those are simple products. Classical RNA is the way we were trying to think about RNA 25 years ago when people were first saying this would be a cool technology to have. And so that is a single target in a single market. That they are important targets. They are emotive targets. They are targets that are challenging growers and beekeepers in specific ways, but they are simple for us to design. Simple, haha. This is a very tough business, very strong technology, but these are the simplest products we can design. And those are the two that Andre mentioned are now in regulatory process. But if I put my old hat on from when I was working in the crop protection industry, one of the biggest criticisms of RNA is, well, it only does one target. What good is one target if I'm just controlling one disease and there's 10 others queuing up to take its place? We need to be able to tackle pest complexes in the field. And that we set ourselves as our generational 2 target. Okay? What we are talking about here is making products that are attractive to farmers and commercial partners. A commercial partner does not want a product that a farmer does not want to buy. And so generation 2 becomes extremely important to us. This is where we are able to seek partners to help us go to market. As you're going to hear, generation 1, very GreenLight focused, and in a moment, Katie will talk about how we will be taking those products to market ourselves. But generation 2 is where we are really able to satisfy needs of farmers and growers beyond those unique situations where one pest, one product is really important. And then we've got generation 3. Generation 3 is the holy grail. It's been whispered of in the halls of some very wealthy companies as they've tried to develop this. And that is to put the RNA inside the plant I'm going to say something now that upsets our formulation and discovery biologists. Any idiot can put RNA inside a plant. Heck, it's full of it anyway, right? But to put it in there and make it functional, that's the trick, okay? And once you can do that, the whole market opens up. As Andre mentioned, the go-to move for a crop protection person, herbicides, okay? But it's more than that. We can start to affect the way plants handle nutrition. We can make them more efficient. We can affect their output traits. We can improve the quality of the proteins they make. We can help them handle abiotic stress, a little bit of water stress or heat stress. We can help them through that with this technology. And so the market, the entire agricultural crop protection market input side opens up to us at that point. And what you're going to hear about today are the successes in Generation 1 and Generation 2, and extremely importantly, in Generation 3. So, shortly after the last Science Day meeting, which, just as a reminder, was on the 18th of October, I believe, 2021, about 15 months ago, we sat down as a plant health team and said, well, how do we take this generational concept, how do we turn that into a strategic roadmap for bringing products to market? And so what we did was we began to overlay the biological barriers for RNA on top of our generational targets. And you can see that when you do that, you get this really nice staircase, okay? You move forward, you increase the sophistication of your RNA design to be able to handle more targets. When the biological barriers, such as the very hostile environment of an insect or the soil for treated seed, those are hostile environments for RNA, because remember, RNA to the rest of biology is food. Okay, it breaks down very quickly because things want to recycle the RNA. They need it for themselves. And so those barriers, as I say, it sets a staircase, and if you tackle each one in sequence, every step that you take, every improvement you take helps you break through the next barrier. And the amazing thing is that we were teetering on the Generation 1 step about, or just starting to think about taking a step up onto that second step there, titled membrane penetration. We were teetering on that step 15 months ago, and now, as I've just described and Andre mentioned before me, and you will hear from the scientists themselves, we are starting to take steps onto that last step. That's a heck of a journey in 15 months, and I couldn't be more proud of the team that has worked diligently to deliver that. Why have we been able to move so fast? I'm going to go back to the manufacturing process that we have. We make very pure RNA effectively and efficiently. And that sets us up. It sets us up for success, because you do not want anything to come between the RNA and the target organism other than what you need it to. And with pure, clean RNA out of a highly efficient and highly effective manufacturing process, that's exactly what we can do. So what does all of that mean? What does all of it mean in terms of the portfolio? We've talked a little bit about the portfolio. What does it look like? Well, you can see our 2 Generation 1 projects at the top. The scope of those has not changed. The progress on this slide from last Science Day to this is the yellow block. So you see, first of all, CPB, we are expecting to be fully commercial this year. We actually, the people that we would normally bring in to a presentation, oh, this is Katie will tell you, are actually in a sales meeting right now talking to potential customers for when this thing gets registered. So we've made tremendous progress. There's a lot of demand, and Katie's going to speak a little bit about that. And our Varroamide product now has taken all of the learnings from our exercise with EPA and folded them into our submission for that product, which has now gone into EPA. Everything else that we were working on back in Science Day has been redefined or has been initiated. And so everything else is now a Generation 2 project. And so you'll hear from Sean in a short while on how our fungal targets are now tackling pest complexes and are not simply powdery mildew on grapes or botrytis on strawberries. You'll hear from Ken about the tremendous progress in being able to control caterpillars in the field. And then you're going to hear from Ron, because about this time last year, we decided that we were doing so well that it was about time we kicked off our journey for a generational free target. And that work has moved faster than we'd ever dared hope. Tremendous progress, very, very excited. And so if you look on the right-hand side of the chart there, you can see something we've not really talked about before, channel access. And you can see what I was talking about on that second slide. Once you move into generation 2, partners become interested. We have been working on selecting partners, and we are making great progress in that area for these technologies. We're very excited. That value, that added value from addressing some of those major concerns that people have had about RNA and showing that they are concerns, but they're not real. Invalidating those concerns. That has opened up those markets for us, and we're pretty well placed. You'll also see that GreenLight intends to continue to participate. And why does GreenLight want to do that? As I said, we need to be technology stewards. We're creating this space. Our work is laying a foundation. And we do not just have to win in the discovery, in the development, in the science. We have to win the scientific backward and forth with the regulators. We have to win the hearts and minds of consumers. That this is a great technology, that this will do everything that Andre said, and it will be tremendous for everybody. And the way that we see that we can do that best is to continue to participate. So GreenLight will continue to be a commercial company in specific markets, in specific places with its Gen 2 and its Gen 3 products. And so the foundation that we laid, the roadmap that we drew, the plans that we made, in 15 months the team has brought those to life, has made it manifest. And as I said earlier, I could not be more proud. The best thing I can do at this point is get out of the way and let the people who know the products, know the projects, and know the technology tell you a little bit more. And so with that, we're going to start with Katie, and she's going to begin with Calantha. Thank you very much.
Hi, everybody. I am Katie Lee, and I have the best job in GreenLight. So I lead our marketing efforts on the plant health side, and in that role, I get to interact with the marketplace. So I get to go out there and talk with farmers, talk with the value chain, talk with policymakers, and hear about all of the excitement for what we're doing. I'm going to talk to you about our Gen 1 pipeline. So like Mark said, these are single target concepts with simple formulations solving unique pain points that farmers and beekeepers have. Now, like Mark alluded to, ordinarily I would have my director of technical sales up here talking to you about how our commercial field trial programs are helping us develop the best possible recommendations for the farmers and give the best possible data-driven advice on how to use what we're working on. Unfortunately, they're in a sales meeting, so you get me. So we're going to talk first about Calantha. Now, for those who are not familiar, Calantha controls Colorado potato beetle. If left untreated, Colorado potato beetle, they're voracious eaters. They will take down a field within a matter of days. Even when controlled, they cause hundreds of millions of dollars in damage, largely because of the resistance that they've developed against many of the actives that are out there. Now, Calantha really represents the promise of RNA in the crop protection market. Now, first and foremost, because it works. We've got 5+ years of field trial data showing that it stacks up against some of the best chemical insecticides, pretty much no matter where you put it. You put it in a field with extremely high beetle pressure. You put it in a field with known resistance, with beetles with known resistance to current actives, not a problem. It also does this at very low use rates. So we're at about 9.4 grams per hectare. Most products out there are about 3x that. So if you're thinking about your AI load on a hectare basis, it's a really nice profile. One of the other things that's kind of been a barrier to adoption for similarly positioned products in the past has been usability. We don't have that issue here. We see that it is very stable throughout the distribution value chain and on the farm. You don't have to use any special equipment. There's no special handling. A farmer can use it just like any other product that they've got on their farm. It also has low to no detectable residue. When we go looking for it, we basically can't find it. Because of all of this, we've been able to develop some really great relationships within the potato value chain. When I go talk to farmers, we've got some really large farms who are really excited about this, given the amount of resistance some of them are experiencing on their farms in the States in particular. So part of getting our product into the market is getting it registered at EPA, who's our regulator here in the US. In consultation with them and some pre-submission meetings that happened a few years ago, we are working with the emerging technology branch within the Office of Pesticide Programs at EPA. And again, in consultation with them, there was agreement to put together a kind of smaller regulatory study program than is typical for other more conventional chemistries. This should allow for a more expedited review program or review timeline. The process has been interesting. It's been good. There's been a lot of robust back and forth as we learn more about how EPA thinks about RNA and the types of data that they want to see, particularly around bioinformatics and non-target organisms. We feel really good about the data package that's currently in at EPA and are standing at the ready for any further questions that they may have. So one of the keys to our commercial activity this year is going to be taking advantage of the experimental use permit that we've applied for. This will allow us to get onto some larger acres and make sure we fully understand how the product works at a larger scale. This will be ahead of a full federal regulatory approval, which we're expecting this year. After we get that federal regulatory approval, we will need to get our state registrations. That's a much different timeline, more on the order of 0 to 2 months rather than in the order of years. This will enable us to be in full commercial activity this year. Full potential for Calantha is global product. So to that end, we are planning on submitting to EU, UK, Mexico, Canada, Ukraine for regulatory approval to help farmers in those geographies as well. Calantha is also protected by a really robust patent estate, both around the AI, as well as the formulation. So all of this together has put us in a spot where we are actively preparing for commercial launch. So part of that is locking down our go-to-market. So over the last few years, we really have contemplated nearly every way that you can get to market. And where we've landed is working with independent distributors who have similar values to us, who really put a premium on having unique products in their offering, as well as having deep knowledge of the local potato markets. As we've gone out and talked to folks across potato country, and you can kind of see from this map that potato country is fairly concentrated here in the US. We've found some great potential partners who we are excited to work with. The feedback from them has been very positive, and we are in talks for exclusive arrangements that will also take advantage of the relationships that we've built to date to go out and co-promote the product as well. So that's Calantha. After Calantha, we have our Varroa mite solution. So for those who are used to more traditional crop protection markets, beekeeping's a bit different. So just to level set, so there are about 100 million beehives globally. really across the world, and that shouldn't come as a surprise as you think about how important pollination is to so many fruits and vegetables and the importance of honey in so many diets. There's a difference between kind of commercial beekeepers, who are the large guys, and hobbyists, who you can think of more as backyard. Even for these commercial beekeepers, they can experience in an average year 35 to 40% loss of their colonies. And when you go out and ask them, and others have formally surveyed them, they say in most geographies, their number one threat is the Varroa destructor mite. And so that's the pain point that we are trying to solve for them. To learn more about the markets, we've been really actively engaged here in the US, developing relationships with key influencers, both in the kind of academic community and some of the large commercial beekeeping operations, and are doing the same ex-US as well, with a focus on geographies where there is a more mature commercial beekeeping market and the developed channels to make sure that we can get our product to them. Longer term, the plan is to go after the hobbyist market as well. It's, as you can imagine, a bit more fragmented. The channels are a bit different, but good opportunity for us there. So the history of this product is that we acquired IP in the late 2020, early 2021 timeframe. And we quickly tried to get off the block with the help of the folks that we acquired from to launch a field trial program. And we did it in key geographies throughout the US to figure out how this worked in our hands. We did use the same form factor that was used before. So for those who are unfamiliar, it's a sugar-filled pouch that a beekeeper puts on top of their hive. They remove a sticker, and then the bees are attracted to the sugar in the pouch and bring it back into the hive. It fits into standard practices. Now, so that was our 2021 trials, kind of figuring out how we make it, does it work in our hands, what exactly do we have? 2022 was about really locking in the rate range based on some of the promising results we saw in 2021. And I've got to say the results exceeded our expectations. So what we see is about 40% more effective mite control over time with longer lasting impact than a chemical control. And this is one of the leading chemical controls out there. So this particular chart is looking at mites per 100 bees. So literally someone's in there counting mites on bees just to see the kind of micro impact. The gray line is untreated control. So unsurprisingly, for something that's considered the number one threat to many beekeepers, populations of mites, if uncontrolled, grow exponentially quite quickly. Now, the yellow line is our chemical control, again, a very, very common control active used today. Green lines are our GreenLight solutions. So what we see at 2 grams per liter is a formulation that competes really well with this chemical control. But what we're most excited about is this light green line, where we're seeing this differential impact in mite control. So what does that mean, though, when you step back and look at it on a hive basis, right? So if that mites per 100 bees is micro, what's that mean for overall hive survival? So we looked at this in terms of hive survival. So this was, last year ended up being a particularly devastating year. I mentioned earlier, it's not uncommon for commercial keepers to experience 30 to 40% losses. In the middle of this trial, we started getting calls from some of the beekeepers that we were working with saying, you guys got to stop this trial. The losses are just too heavy. We had a hunch of why that was, largely because we had an untreated control in there. But the losses that they were seeing were on the order of about 75%. when you are making your money off of, the number of hives that you can deliver into pollination services, the amount of honey that you can produce, having this level of loss really hits your bottom line. So that's where we're so excited about the observation that we saw at the mite level holding at the hive level, because this green line is really showing great results in keeping these hives alive over time. One of the questions that we're going to keep asking ourselves is, how does this actually translate into hives of bees? So that's one of our key questions as we move here now into 2023. One of the other questions we asked ourselves, we kind of had a hunch here, was how does this work? We want to make sure that we can tell our customers and understand ourselves, what is responsible for this, especially this longer lasting impact that we're seeing. Most of the products out there right now impact phoretic mites or kind of more adult mites that are outside of the cells. What we are seeing is that when a hive and a cell has has our varroa mite solution, the number of mite offspring is dramatically decreased. So actually, the researchers, some great collaborators we're working with in New Zealand, basically saw no daughter mites when they went looking for offspring. Now, when you have the solution in a capped cell, so in there with the developing bees, we also wanted to be very sure that we're not impacting the larval development of the bees. And that's what we're seeing too, is no detectable impact on bee people mortality, which is a really important finding for us. Now, in order to move this forward, also regulated here in the US under FIFRA at EPA, the submission was just made last month. We were able to take a lot of the learnings that we had throughout the Calantha process and feed it into here. But again, like Calantha, full potential here is geographic expansion. So we are looking into Canada, New Zealand, Europe, UK. Again, some of these more mature beekeeping markets with a number of others under consideration. The patent estate for the varroa mite solution is also very robust with patents and pending applications covering the active ingredient, gene target, as well as the field of use of dsRNA against varroa. So that's our Gen 1 pipeline. We are really excited by the promise that it shows in terms of effectiveness as well as safety profile, and are looking forward to bringing this into our second and third generation products. And Dr. Sean Whipple here is going to talk to you about the great work that he is doing to develop out our Gen 2.
Thank you, Katie. Andre said we were going to nerd out on you guys, and here I am. So, no, my name is Sean Whipple. I'm Director of Global Technical Development, basically the field guy. And as somebody who has devoted my life to agriculture, crop protection, really exciting stuff to be here talking to you about. Multi-target solutions, I'll get right into the first one, which is powdery mildew. Now, when you think of powdery mildew, most people probably think of grapes, first and foremost. And that's a noble goal. $1.4 billion addressable market. The average grower in grapes is going to spray 7 times. It's a very high input crop. So if you think about a high producing state like California, 74% of their inputs in grapes are specifically targeted at powdery mildew. That's 17% of California's total pesticide load just targeted at powdery mildew. So it is a big market. However, there are closely related, genetically similar species of powdery mildew that really expand the utility of a product for a grower. So with expanded targets also comes expanded value. And If you think about back to the 90s, the introduction of quinoxaphene, the first specialist powdery mildew product that could control powdery mildew in cucurbits and grapes and vegetables, it becomes a very useful tool in agriculture. So that's exactly what we wanted to do. We wanted to modify our lead sequences to not just target grape powdery mildew, but to address issues in cucurbits like Potosphaera and cereals with Plumeria, and have a real solution for growers to cover powdery mildew. So moving on, kind of a where we were versus where we're going and where we are story. We knew we could control powdery mildew in grapes. We knew we could show gene knockdown in grapes. If you look at the table in the lower left, two of the sequences there, PM1 and PM2, are providing roughly 70% control on average. That's equivalent with the biological standards that you have in the market. Keep in mind, though, that is without formulation. That's without optimization. That's naked double-stranded RNA sprayed in the field. It is capable of doing that. When you look at a conventional control, that is generally providing around 90%. Now, you're going to see a lot in my section where we talk about industry standard, chemical standard, biostandard. What I can tell you is that we benchmark against grower standards, what growers are using in the field and what they would put out under heavy and normal disease pressure. And we knew that, and now we know that we can control cucurbits. This is Potosphaera. This is a completely different species, different genus of powdery mildew, and in one sequence we're able to control. If you look on the left, that is 60% disease pressure, incredibly high disease pressure in the field. And we're able to knock it down 2 levels, almost on par with industry standards. If you look at the photo on the right, I have to plug our internal research station that we have in Spain. This was done at our own facility. And we utilize our Spanish research station as well as our controlled environmental research team here, which many of you will see on the tour later, to rapidly progress our targets and our sequences into field development. So we tested these 6 candidates, and roughly all of them are comparing very well for their control of cucurbit powdery mildew. But that is naked dsRNA, no formulation, no optimization. If we look at optimized dsRNA, now we have a formulation. We know how to make it bioavailable, and we know how to make it more applicable and more valuable to a grower. You can see that PM1 and PM2, the same sequences, when formulated and optimized, were able to drastically reduce disease severity in cucurbits. This is validated in both the zucchini as well as melons. So again, photos of very high disease pressure, and our sequences there able to then provide equal or even greater control than the chemical standards that are available. You can also see that with the advent of adjuvants, adding adjuvants to it, we can increase the bioavailability of double-stranded RNA to the disease. So that's an organosilicone there, and you see the AI loads of the industry standards, 470 grams, 290 grams. We're working with 20 grams of active ingredient of double-stranded RNA to achieve that level of control. If you look at continuing to expand this spectrum, this is testing in wheat, and early results show that we have very good control of wheat. Cereal powdery mildew, 6th most economically damaging plant pathogen globally. Very, very important disease. And I can also tell you that this industry standard in the middle, after 15 years in the industry, that one in particular, if you can compare to that on wheat powdery mildew, you have a product. And very exciting for me to see starting to work with double-stranded RNA in the field. This is naked dsRNA providing that level of control without optimization, and we think we're going to be able to do exactly what we did in cucurbits, be able to optimize that level of control to be even more viable to a grower. That being said, our sequence PM1 that you saw, we are elevating into full-scale development. And what that means is that the regulatory dossier data package will start to be developed for the EPA within 12 months and subsequently 24 months to the EU. During that time, we will do what we call optimizing the gap, that is good agricultural practices. How is a grower going to use this? well, how does he need to spray it? When does he need to spray it? What does he need to mix with? What is the application timing? So doing all the field tricks in order to understand how to convey that information to a grower. So we will be doing that over the course of the year with a targeted submission of next year, 2024, for this powdery mildew product. That is concurrently pending U.S. and international patent applications covering our powdery mildew double-stranded RNA AIs. Moving forward into gray mold, something that I'm assuming many of you are familiar with. If you've been to a grocery store, you have probably seen some botrytis, but gray mold plus. The plus is important in this project. So with gray mold, it causes roughly 30% loss of all global food production. It's a very serious disease. It's ubiquitous. It occurs everywhere, and it occurs on over 500 species of plants globally. So when you think of that, botrytis being so serious to spoilage of produce and fruits and crops, we want to control botrytis, obviously, but we also want to control other diseases that occur at the same time. One of the biggest problems with botrytis is that it's incredibly aggressive. And by the time a grower may notice that it's there, it might actually be too late. So it's high input, multiple sprays, and if it gets to the point where it's severe, there's very few things that are available on the market to actually control botrytis and control it well. So we want to target botrytis, but we also want things that occur at the same time that are genetically somewhat similar, like sclerotinia. So what we knew we could do is use naked dsRNA in the field to show gene knockdown. We knew we could control botrytis in snap beans and also in strawberries. But if you look at those numbers, the percentage of disease control, you're probably thinking 50 to 60% control isn't that great. For a disease as severe as botrytis, there are very few compounds that provide exceptional levels of control of botrytis because it is so severe. So 50% control, 60% control in a grower standard rotation is commercially viable. And this is with naked double-stranded RNA. But we obviously want to increase that. We want to optimize that and bring a tool that is all both useful and valuable to growers. This is some work that we did, again, on naked double-stranded RNA in the field. And if you look, screening different sequences that we have available, BC1 through BC4, we screen these in Spain and in the field using contract researchers and use our CER team to figure out where our sequences play versus these industry standards. But I want you to focus really on this red dot in the upper right. That is what I like to refer to as a kitchen sink compound. When the grower has no options, a super serious disease pressure, he throws the kitchen sink at it. That's a kitchen sink compound. So very popular and well-respected industry standard chemical control. So if you see BC3, one of our sequences there, from 10 to 40 grams, we're seeing that dose rate. And up at the higher end of the rate range, we are equivalent to commercial standards for botrytis control. That's incredibly exciting. Some of the sequences, obviously, that provide lower levels of control to the biostandard or little to no control or eliminated from the screening process, and we move BC3 forward into the field. When we do look at BC3, you can see under pressure from both botrytis and sclerotinia, it is providing good levels of control. This is with naked double-stranded RNA. We're controlling both botrytis and sclerotinia at the similar application timing. Now, what you'll notice is the three segments down the center of the graph, we're not just looking at the spectrum here compared to the accepted chemical standard. We're looking at actual grower application strategy. And I'll tell you what this means. 7-day spray interval, when you have very serious disease pressure, the growers want to keep those sprays tight. They want to keep them close together so that they can tackle that aggressive disease, more input, more sprays. Now, if you can expand that to 10 days, 14 days, that's one less time a grower has to get into the tractor. That's one less time he has to go into the field. That's less fuel he has to use. It's less input. So it's very important to know that we can take double-stranded RNA, use it on a 14-day schedule, and get equal performance to what a grower would use on a 7-day schedule. So this is with naked RNA. We're seeing equivalent control of BC3 at 20 grams AI per hectare, 7 days, 14 days, controlling both botrytis and sclerotinia. And we anticipate that with optimization, similar to what we did with powdery mildew, we'll be able to increase that level of control even further. And with that, we're working to elevate BC3, that sequence, to full-scale development, most likely in 2024. When that happens, we will do the same thing as powdery mildew. We will start building the dossiers for the EPA within 12 months of that happening, the final formulation, and then within the 24 months to the EU. During that time, we identify the gap, grower use patterns, how they're going to use it, write the label with the first submission planned for 2025. So I appreciate your time just talking about these two fungal projects, but with that, I'm going to turn it over to my colleague, Dr. Ken Narva, Head of Insecticide and Fungicide Discovery for GreenLight, and let him talk about some Gen 2 discovery programs. Thank you.
Thank you, Sean. Hope you all enjoyed those field results. That's really special to see and really great to see the progress of things that have moved out to the field in the last year. So today I'm going to tell you about 3 projects that are further upstream in our development pipeline. We call these discovery programs. And so what goes on in discovery? So you heard about the green box. That's where it all starts. We work with our data science team to look at transcriptomes and genomes and come up with targets that are sensitive to double-stranded RNA and RNAi, and things that we can then develop as insecticides or fungicides. That all happens in silico and then into the lab. We go from the lab to the greenhouse and to the field, and so that's the progression of some of these projects that I'll show you today. First one that we'll talk about is mycotoxin control. And mycotoxins are a small molecule toxin that has severe gastrointestinal disease symptoms in man and mammals, produced by a number of different Fusarium species, particularly a problem in wheat. So globally, probably a $5 billion or more opportunity to reduce the amount of mycotoxin in wheat. As you can see there on the global map, this is a problem worldwide, and simply the contamination of wheat with mycotoxins renders it useless to the grower and to the downstream producers. So in the laboratory, what do we do? So this is a workflow. So imagine now that we've talked to our data science team. We have a number of different sequences that are candidates for controlling directly the DON production. Most of the current fungicides that are used are non-specific for control of Fusarium, but in this case, we're specifically targeting DON production. And we have come up with a high-throughput screening method whereby we can grow in liquid culture various DON strains and impact them with double-stranded RNA. Being able to do this in high-throughput format gives us the ability to look at different delivery technologies, different growth conditions, and then measure DON production by HPLC chromatography. So far this year, going back in last year, we've looked at almost 300 strains, or 300 sequences rather, in this system. We have now 40 that are performing well in an in vitro detached leaf head assay, and we anticipate to get 15 of those into the greenhouse later this year. So that's sort of the progression of hits and the rate of advancement that we typically see in these screening programs. So here's a little bit of data for you to look at. We're looking at DON reduction now with some different double-stranded RNA candidates. So the left-hand panel is the percent DON reduction, achieving about 80% in this liquid format assay. So these are what we call hits. And so we'll take those forward in the pipeline and evaluate them further. On the right-hand panel, that's the relative expression of the target genes when we treat Fusarium in this liquid assay. So gene knockdown showing that the mode of action for this DON reduction is indeed RNAi. So as mentioned, the next step then is to get that onto some plant material. So we have this detached leaf head assay that we use in the lab, where we can treat with Fusarium infested onto those wheat heads with double-stranded RNA. And again, measure DON production or reduction in this case by our double-stranded RNA. So the panel on the left, you see the lower dark green bar, the impact of reducing DON production with RNAi. And then on the right-hand side, some pictures of the reduced disease symptoms that one sees in these wheat heads. So good progress with this project. We think that when we get to the field, we may anticipate the need for some additional uptake improvements. The results I showed you were just using naked double-stranded RNAs. If one labels RNA with a red dye that is visualized by confocal microscopy in the fusarium mold, you can see on the left, we don't really get a lot of uptake with naked double-stranded RNA. Yet you did see we had a phenotype. So we're on a good path, we're making progress, but we're already anticipating that if we can enhance this, it will improve our cost of goods. So using different agents that promote the uptake, you can see the panel on the right, there's a lot more red labeled double-stranded RNA in that fusarium that's been treated with some delivery technologies. So that's always an active area for us. Start with naked RNAs and think about how we might improve upon that activity by adding in some delivery technology. So now we'll move on to another project that's also in early development, and this is for plant feeding mice. So we speak of multi-target opportunities. This is a classic example. The picture you see there is two-spotted spider mite, but it is just one of a complex of mites that affect a large, wide range of crops, especially high-value crops, fruits, nuts, vegetables, both in the open field and in the greenhouse setting. So it's a very, very big problem. Also a big problem because it's prone to developing resistance. These pests reproduce very, very quickly, and so with high application rates and frequent application rates of various acaricides, you see now that the spider mites are often resistant to up to probably 90 some odd different active ingredients in the field. So RNA as a new mode of action has an opportunity to overcome resistance in these populations of mites. One thing that's really appealing about using RNA for a situation like this where you're spraying on fresh consumables is the low residue, quick disappearance, as Katie mentioned earlier. The low residue profile for RNA fits very nicely into these consumable markets. So data here I'm showing you now, sequences that we brought in from a collaboration that we have. And on the left-hand panel, what you're looking at is mortality curves, or a decrease in alive or vital mites. So by 9 days or so, treating with the two sequences we're looking at, you see a rapid decrease in the green lines of the spider mites in this assay. Meanwhile, water or an untreated control, the numbers are much, much higher. Also important in this concept is to reduce the overall population. So we refer to that as fecundity, and so we're able to reduce the number of eggs or fecundity with this double-stranded RNA controls. So on the right-hand upper top panel, you'll see there that the two different sequences we're working with have given a very large percentage of reduction, over 80%, compared to the controls. And the commercial standard, of course, this is a finished product, does a very good job in this assay, but again, that's a finished product, so that's what we're benchmarking against. Bottom right panel, again, looking at RNAi knockdown of the target gene, and you can see there that indeed the reduced expression of each of those targets indicating RNAi as the mode of action. So in the laboratory, then we go from doing some artificial assays like I just showed to work on plant material. So in this case, we're using detached leaves that have been treated with the double-stranded RNA. We're using mites that have been pre-treated and then placed on these leaves. And there's 3 different trials depicted there, left to right, with water formulations, negative control sequence, and then our 2 target sequence is boxed in red. And you can see whether the reading was at 27 days, as was the case for the top trial, or 15 days. You should be able to see some yellowing in the control water and the negative control sequence. You don't see that yellowing, which is the chlorosis caused by the mites in the two RNA sequences. So good protection in this laboratory assay. As always, things in the lab look good. You've got to translate that to whole plant material in the greenhouse and then onto the field. So this is work that was actually done by Sean's team in the greenhouse, same sort of situation, now spraying on whole soybean plants every 7 days or so for the duration of the trial. During that period of time, the plant is growing and it puts on what we call trifoliates, new sequences of 3 leaves. And so from the very left-hand side, those are the first leaves to appear, but they're called, in this case, the oldest trifoliate. And you'll see the upper left control untreated yellow chlorosis, as we saw a little bit better in the previous slide. You know, moving on to the newer trifoliates, you'll see a little bit less damage, but still damage on the untreated. Our double-stranded RNA sequence does a very good job of cleaning up the mite damage, and you see nice green leaves, somewhat comparable to the commercial standard in the bottom panel. So we're making really great progress with this project, getting ready to file intellectual property, and we should be into the field later this year with this program. So now we'll move into an area that we've been working on for 2 or 3 years now, and it's with Lepidoptera. And Lepidoptera, worldwide, when you look at the wide range of leps and chewing caterpillars that affect multiple different crops, this is a multi-billion-dollar problem for growers. We're going to talk about diamondback moth as one example because it is very important in the vegetable market. But really, when we think about Lepidopteran control, we're thinking about controlling a complex of organisms. It's a whole spectrum of leps. So back to the idea of multi-targets. In this case, we have more than one Lepidopteran that we're going to need to control. We're going to do that by building in by design, both specificity but also spectrum. So we want to keep it narrow and safe in terms of the spectrum of organisms that are impacted, but we want to increase the spectrum so that we can bring the whole range of Lepidoptera into control. So I want to give you a little bit of background about why Lepidoptera are so difficult and why folks in academia and in industry have been really unsuccessful in being able to control leps with double-stranded RNA for over a decade now. And it's all due to the fact that Lepidopteran larvae are built to eat and digest. So RNA, which is part of their normal diet, is digested very effectively by their midgut. On the left-hand side, I've called out some of the challenges in the midgut environment. And then there's a cutaway segment of in this case, a fall armyworm, but it serves to show the purpose of the architecture of the midgut from the foregut to the midgut and hindgut. And then there's a cartoon blow-up of what goes on at the midgut level. And it's important that you understand this. So when RNA is ingested by the Lepidopteran larva, the first thing it encounters is a midgut that's full of RNases. So that's normally there to digest the RNA that's in the plant material that would be the food. So naked RNA is not going to survive that midgut. There's also an alkalinity of the midgut that is a problem for RNA. Higher pHs can cause some degradation of the RNA. Even when it's intact, then the RNA has to traverse the peritrophic membrane. So there's a thin layer that lines the midgut cells from various food particles and other agents that are ingested by the insect. Okay, assuming one then can get RNA beyond the peritroph, it then is taken up by endocytosis into the midgut cells, which are those boxes. And at the cell lining, it's taken further into the cell and trafficked through the endosome to the lysosome for degradation. So all those steps along the way, from being ingested to degraded by nucleases to barriers, physical barriers to uptake, and then cellular degradation, all have to be considered in trying to address LEPs with RNA. So we focused a lot of our attention on how to accomplish that delivery. And this is a pretty simple slide to look at, but there's a little bit I want to talk about to you in what's in the middle of the graph. So we've come up with a delivery system that's built on naturally occurring biocapsule that has no activity on its own. And we found ways by partnering with people at the University of Massachusetts who work on RNA therapeutics for different drug delivery purposes. We've come up with a mechanism whereby we can stabilize and load RNA into these biocapsules. Now this is a really important advancement. It gives us the ability to put RNA in, stabilize it, then tune its release for when it hits the right physiological environment in the insect gut. So that's a clear advantage over some of the microbial production systems that you'll read about that might be in other companies' platforms. The ability to tune these exactly the way we want is really important. And the graph shows actually some data from Sean's team where we did a field trial with these encapsulated RNAs on diamondback moth. And on the very left-hand side of that graph, you'll see the untreated control. And what's being measured here is defoliation, so the amount of consumption of leaf by the insects. So you see about a 40% defoliation, and that's a typical field rate of damage. And you see the commercial standard is the next light green bar. And we're doing pretty well with our sequence at two different rates in the dark green bars. So it's a relatively high rate of application in this first trial, but I think this is one of the first examples of controlling Lepidoptera effectively in the field with RNA, in this case delivered by our delivery platform. Even more exciting, I would say, in working up the delivery system, we made a discovery that the biocapsule plus certain coformulants can exhibit an increased activity. So this is a little bit of a complex slide. What it shows is in the light green bars, various treatments that don't have any biocapsule. And then in the dark green, increasing concentrations of biocapsule along with increasing concentrations of this coformulant. So coformulant alone, the first three light green bars, you see there's very little activity there. And then you add in more coformulant and more biocapsule, and you get very good protection against defoliation. So this was really exciting to us because now it suggests that not only do we have a platform of delivery where we can deliver RNA, we can begin to add in other components to further enhance our overall activity and pest spectrum in the field. Another attribute of the system is its ability to reduce the dose response in resistant insects. So you heard earlier that one of the main things along with residue and registration was to be able to overcome insect resistance. So this is work that's been done in the lab with this particular strain of diamondback moth that's resistant to insecticides. A little bit of complex data here to look at, but on the left-hand upper panel you'll see the biocapsule alone, and there's really no mortality there against the DBM. And then on the right-hand part of that graph, as you increase biocapsule with the coformulant, we're achieving up to nearly 100% mortality on an insecticide-resistant diamondback moth. So that's a really important discovery for us. If you graph that out and just think about it, on the right-hand panel, there's a dose-response curve shown in gray. So if you take this coformulant and do some dose testing and then add in the biocapsule, you shift that dose response curve to a more sensitive rate that can be close to 2 orders of magnitude. And then on the bottom part of the graph, if you express that then as a resistance ratio, we've lowered the resistance ratio, that is increased the sensitivity from a calculated number of 151 in this case to about 12. So over 10-fold reduction in resistance ratio or increase in susceptibility. So now we've got a platform. We can deliver RNA, we can load it, we can tune it, and it's got attributes of overcoming resistance. This has also been shown here in the greenhouse, kind of another intermediate step in the way, but increased biocapsule in dark green, along with coformulant at increasing rate. You see the decrease in defoliation. So more examples of plant protection, and then some pictures on the inside on the right, untreated control top right or top left, coformulant, some damage on the middle, and then the biocapsule plus coformulant is essentially clean from feeding damage. It might be a little bit hard to see, but that's if you think about the dark spots on the top untreated control, that's what you see a reduction of and an elimination of with the biocapsule plus coformulant. So last slide for me, I'll take it back to the beginning, the field data. You know, first demonstration of double-stranded RNA controlling Lepidoptera in the field. Underpinning that is RNAi from a particular sequence that we've worked on for a while now. It's got really good lethality when one treats diamondback moth cell lines with it, and you look into that further and you can see the reduction in gene expression indicating RNAi is the mode of action. So we've come a long way in the last 2 or so years with our LEP program. It's now got the ability to be broadened out beyond diamondback moth. We've shown these same sort of results with fall armyworm, and so we'll build out spectrum as we go forward with this program. And so now it's my honor to introduce Ron Flanagan, Vice President of Plant Health Discovery, to talk about our plant delivery program. Ron.
Well, it's my pleasure today. I get to talk about plant delivery, our Gen 3 products. And something I would want to point out is, you know, Mark talked about where we've came in 15 months. 12 months ago, 50% of this team was not at GreenLight. So this team has done amazing work along with a cooperator or a collaborator, and been taking results from the lab to the field in less than 12 months' time. And you'll hear about that work throughout the presentation today. So first off, you know, enabling plant delivery unlocks significant potential opportunity for GreenLight. And when people think about plant delivery or delivery, they think about foliar applications. And of course, what leads the way with foliar applications, which is roughly a $37 billion market. It's dominated by glyphosate. Its traditional chemistry is under pressure due to resistance development and also from the regulators, not only in Europe, but in the US. There's a limited number of AIs in development. And actually, some of the most recent technology that's come out is using AIs that were discovered 50 years ago. Extend is a great example of that. Then you have your seed treatment market, which is about a $6 billion TAM, and it's expected to grow at approximately 8.4% through 2030. Again, some of the things you can impact with the seed treatment, some of those early seedling diseases. So you hear about fungal protection on some of the early seedlings or damping off on early seedlings causing a problem. Again, this is the potential for RNA to come into this space and help provide protection in those areas. So I want to talk about the seed treatment opportunity first. So again, we're working with a collaborator here, Germain Seed Technology Incorporated. What we've done is provided double-stranded RNA targets to Germain's. Germain's has then formulated them in their standard seed treatment process, applied them to seeds, and then they've sent them back to us to look at RNA stability throughout that process. And then we're doing some bioefficacy testing on those seeds as well. So you can see at the top of the slide here, we have kind of our standard treatment. And at the different A, B, C, D, and E, or A, B, C, D, we took samples. We made sure that that RNA was stable throughout the process. And I really want to focus on the dry seed. And the interesting thing here is not all seed treatment applications are similar. What you have in the mix for lettuce might be different than what you have in the mix for spinach. And of course, knowing that, we had to test and make sure that there was stability across both of those processes. And as you can see in the left panel at the bottom with lettuce, you see that the double-stranded RNA we've tested in 2 conformations, naked and complexed with a delivery technology. And the naked RNA is degraded as it moves through that seed treatment process. But when we complex it with a delivery technology, we can then stabilize it and it survives that seed treatment process. However, if you look at the panel on the right in spinach, naked double-stranded RNA survives that spinach seed treatment process just fine, just as well as the RNA complexed with the delivery technology. So now that we know that we've got stable RNA in both of these seed treatments, we want to try to understand, do we get an impact on the plant? So the first thing we wanted to do is look at gene expression changes that were triggered by this seed treatment. And the way we're doing that is using the innate systemic acquired resistance pathway, which is an inducible defense mechanism that the plant uses to induce physiological changes to help combat whichever pest or pathogen it's fighting. So when the plant senses a pathogen, it triggers the pathways, and then it has the physiological changes impacted. There are master regulators or control elements for each of these pathways. However, when the fungi is sensed, these regulators are then changed and no longer able to act as brakes on the pathway, and the pathway is then activated. We're using a similar approach by downregulating the master regulator in the presence or absence of pathogen, and then seeing if we can stimulate the pathway. So if you look on the panel to the right, you can see the target gene. Our target gene is the master regulator that we're providing RNA to target. And then the next 4 bars are actually markers of the systemic acquired immunity pathway that have been activated. So we've downregulated the target gene. We've shown that downregulation of that target gene actually does kick on the SAR pathway. And now what we'd like to do is say, okay, so we've got the pathway on. What does that lead to in a biological response? So I'm going to switch gears and go to spinach here. So we've got some data from spinach that addresses that, or from lettuce that addresses that question. So we did do a chamber test with treated lettuce, and we did see significant biomass and vigor increases in RNA-treated seedlings. So the gray bars are the standard treatment, and the green bar is our treatment CT1. And by 27 days on the left panel, you see roughly a 25 to a 30% increase in biomass. And then the right panel is an agronomic rating of vigor, and you see that increase going from a 4 to a 6. And from my time that I spent in the field looking at row crops, a 2-fold increase in vigor rating is very noticeable by the grower. And actually, it was so noticeable by our folks at Germain's, they were so excited, they wanted to push this to a field trial in Yuma, Arizona at the end of 2022. So we were able to get this material into the field in Arizona. And here are the results from that trial. We did see increased lettuce survival under a fusarium challenge in the field. Again, the green bars are RNA entries. Significance is denoted by an asterisk. So in the first panel there, you're looking at the variety tamarack. And you can see our RNA treatment as well as their lead development candidates are statistically better than what they're considering the untreated control. And what they mean by untreated control is a standard seed treatment without any fusarium AIs, without anything targeting fusarium. But then if you move on to the El Guapo variety, you see that CT1 has a significant increase in harvestable lettuce plants compared to CT2 compared to CT1 and their internal development candidates. You see similar results then as you move to other trials or another location in the different varieties. So again, you see some significant increase in harvestable plants. And the other thing to note here is that CT1 and CT2 are actually different delivery technologies. So we're showing that complexing our RNA with different delivery technologies, we can also get different physiological responses. We can get better physiological responses with some than with others. So as we talked about, foliar delivery is really critical to unlock for GreenLight. And one thing, a foliar application allows for tissue and temporal manipulation that you currently can't get to today with traditional synthetic chemistry. So some of the things you can do is you can spray on in advance of a drought, can potentially spray on in advance of a drought condition to change the plant in such a way that it's more drought tolerant. Another option is you could increase seed size in a plant so you can increase yield. Another opportunity may be to spray and allow flowers to be retained longer to extend the pollination timing. There's also the opportunity to better use nutrients. And then, of course, you've got the largest opportunity of herbicides as well. So the first thing you have to be able to do is deliver to a plant to unlock any of this value. So the first thing the team started to do is look at a model organism and start looking at delivery technologies and the ability to suppress an exogenous marker gene in a model system. So the top panel there is green fluorescent protein, and the interesting thing here is under this wavelength of light, green fluorescent protein shows up as blue. So the first panel is a control plan, and as you can see, you see the blue evenly dispersed throughout the plant leaf. And in the second panel there, you see that there's black spots on that leaf. And those black spots are actually a lack of GFP. So approximately 100% of those plants, when sprayed with RNA complexed with this delivery technology, showed this phenotype or some resemblance to this phenotype. So we wanted to make sure that that phenotype was caused by gene downregulation. So the panel, the bar graph off to the far right there, is just looking at GFP reduction at different rates of double-stranded of RNA that's sprayed. We next said, so okay, well, we can do that in an exogenously applied leaf or exogenously applied transgene. People have said that that's easy to do. So of course, the next step is we got to move to an internal gene, and the team chose CHLH, which is involved in chlorophyll production. So if you knock out or delete CHLH, you do not get chlorophyll and you get a splotchy or you get a yellowish green phenotype on the leaves. In the first panel there on the bottom, you see that an untreated leaf is green, looks normal. And then you can see the next panel is a treated leaf with the RNA targeting CHLH complexed with the delivery technology, and you see that splotchy phenotype caused by a reduction of CLH and the lack of chlorophyll being produced in those areas. So this is really exciting, really lays the groundwork for optimization and further development, not only on the herbicide, but also in some of our crop improvement traits as well. So Andre already kind of stole my thunder, but I'm gonna put the slide back up. We do have some preliminary data to show that we've taken that CHLH and we've parlayed those results into weed control. So this is a picture of horseweed. You have an untreated horseweed on the left, And then you have 2 treatments, CT3, CT4 on the next. And really those are different designs of RNA complexed with the same delivery technology. So again, we've got different designs. We can pull the design lever to get better activity. We can pull the delivery technology lever to get different activity. And this data was backed up by some image analysis, which is showing a significant reduction in biomass with CT4 compared to the non-target control. So again, preliminary result, working hard to build on this result to get to an improvement in what we see here. So I just want to summarize with my final slide on our accomplishments to date. In the seed treatment area, we've demonstrated RNA is stable through the seed treatment process, and that is in naked RNA can be stable as well as RNA complexed with delivery technology. We've shown that in both lettuce and spinach. We've demonstrated in-plant activation of the SAR pathway through seed treatment of RNA. Again, that data was with spinach, showing we were able to downregulate the target gene, which then led to an activation of the pathway and an upregulation of the response genes. We've demonstrated that seed treatment delivery in chamber and field conditions leads to an increase in plant health, as well as an increase in harvestable plants with respect to lettuce. And in the foliar delivery space, we've shown that we can deliver RNA to knock down an overexpressed exogenous gene. And then we built on that to show that we've also been able to knock down an internal gene and provide a visible phenotype. And as I mentioned earlier, the successes in those areas with those delivery technologies, we've been able to build on and start really getting our weed program, some great results in our weed program. So congratulations to the team. We've got, it's just a testament to the dedicated people we have here at GreenLight, how much they've done in 12 months, less than 12 months when they're highly motivated. So at this point, I'll turn it over to Andre and he'll wrap up.
Thanks, Ron. So as Ron said, there's just no limit to what a highly motivated team can actually deliver. When we started the company now 15 years ago, we started with a simple premise, which was, can we make biological molecules that are far less expensive than what we have been traditionally been able to make, and can we use those biological molecules to start systematically replacing chemicals in our food supply? That was the original premise of GreenLight. That is why we exist as a company. When we went out to the investment community, to the scientific community, they told us that that was impossible. 2 different Nobel laureates told us that synthesizing RNA in a cell-free process where the end result would be a scalable, inexpensive RNA of high purity that could be gainfully used to replace chemical pesticides was impossible. Not 1 laureate, 2 Nobel laureates. Within 5 years, we invented a new way of manufacturing RNA, which today has been scaled up to metric ton quantities in our Rochester facility. We demonstrated that even though 2 separate Nobel Prize winners, by the way, this weren't people who won their Nobel Prize in literature. That would have been funny. No, these were 2 of the people who have established the fundamental mechanisms by which RNA is transcribed and translated. When those folks tell you that what you're trying to do is impossible, you kind of have to listen. Thank God we didn't, because we demonstrated that what other people thought was impossible was just a matter of resiliency, of creativity, of innovation, of inspiration. And we demonstrated that that was, in fact, possible. We then were told, not by 1, not by 2, but by 3 major agrochemical companies, that RNA was never going to work outside targeting of Coleoptera. The potato beetle that we talked about today and will be our first commercial product has historically been the most conventional target for RNA because it's relatively easy to address. Now, nobody had been able to address it with RNA that would be scalable at a commercial manufacturing process, But the common knowledge was you can kill potato beetle with RNA. And so we did, and in the process we carried out 5 years of field testing in 14 countries, where every single trial pretty much looks like the next one. Dead potato beetles, rescued potato plants. No residue on the food, conventional application rates. But we were told that we were never going to succeed going beyond potato beetle. And that in fact, a wide spectrum RNA or a controlled spectrum RNA, an RNA that could kill more than one pest or control more than one pest, was in fact impossible. What you saw today is not one, not two, but multiple demonstrations that we can use a single RNA trigger to control multiple fungal species. We were then told that RNA was only going to be ever applied as a foliar spray and was only ever going to be used to control things that would rest on the leaf and feed off the leaf. That we were never going to succeed at getting RNA to be a systemic active. That RNA was never going to be able to be coated onto a seed because the soil contains too many microbes that will degrade your RNA. That you were going to be limited to things that could be sprayable. That delivering RNA systemically to a plant at commercial rates would be, again, impossible. Today you saw, not one, but two demonstrations of RNA coated on seeds, controlling pests that cause tens of billions of dollars of annual damage and up to 500,000 deaths per year on fusarium. Coated on a seed with a commercial process. Finally, we were told that spraying an RNA and either introducing a trait or creating a highly specific herbicide with no lasting impact that was not going to accumulate on the food was, again, impossible. Now, I don't think that we have conclusively proven that we will deliver an RNA herbicide, but I think that we have proven that we can deliver RNA through a sprayable and have it be systemic in the plant. Just think about the possibilities. Think about the ability to not have to have genetically engineered crops to be herbicidal resistant. Think about every crop in the world being GreenLight ready. Think about a no residue future where you don't have to worry about your kids being allergic to a particular chemical that we're spraying into foods. When you think about all the possibilities that a sprayable systemic RNA brings, drought resistance, higher productivity, higher protein content, the ability to grow crops in hostile conditions. All those things that we were told were impossible, we have systematically tackled. And we have systematically tackled because we are rigorous in our science and relentless in our pursuit of the goals that we have as a company. As we move from where we are today, and we have started climbing this staircase that Mark talked about, from growing from the very simple foliar delivery, to being able to target multiple species, to being able to garner the interest of the major agricultural companies whose pipelines are now running dry, being able to coat seeds and pretty soon to be able to spray onto crops. As we move up this ladder, what we envision is a world where we achieve the ultimate impossible. The ultimate impossible, if you ask world experts, is they tell you it is impossible that we will be able to feed 10 billion people with the land that we have devoted to agriculture today, that we have to have more land. And if we have more land, there is no way that we can mitigate global climate change. That is the sentence that has been given to humanity. You will either starve or you will boil to death. And there's no way around it. A solution to feeding 10 billion mouths while sustaining our planet, while being able to have agriculture that does not fundamentally change our biodiversity and our ecosystems, is impossible. And to GreenLight, there's nothing that gets us more excited than when somebody tells us that something is not possible. Because then our minds and our hearts and our souls get focused on achieving the next impossible. And that's the next frontier for GreenLight. Now I'll give you another impossible, right? Which is raising capital in these crazy times. Many biotech companies are finding that impossible. At GreenLight, for us, it's just another challenge that we will overcome. For me, it's truly an honor and a pleasure to be able to share with you the progress of 12 months. Think about it. 12 months ago, we had one product candidate in registration. One. Today, we have product candidates in the field, in registration trials, addressing multi-billion-dollar opportunities. And we have unlocked the key to systematically changing how agriculture gets done in our planet. So I want to thank everybody for your attention. We have over 100 participants, both in person and online, and that's truly a pleasure to see so many people interested in achieving the impossible. I think we have a few minutes, and so if anybody has any questions here in the room or online, we will be delighted to try to answer them.
Sorry about that. Thank you. Thank you for the presentation for everyone. So I have a couple of questions. First one is that how do you address the resistance to RNA? Because you have a resistance coming up with insecticides and biologicals and so other modes of action. So pretty much an RNA is not that, it's exempted from that because it's a biological molecule like the other ones, like protein. Even protein gets, resisted by the insects and pests like fungus and even viruses. So how do you account for that?
Yeah, so the question is, how do we address the upcoming resistance to RNA? And we agree with you. I don't think that it's a matter of if we see resistance, it's a matter of when, right? Nature finds a way, as we learned through Jurassic Park. Nature will figure out a way to get around methods of control that we have. I will submit to you that an organism figuring out a way around RNA is going to be more difficult than figuring out a way around a chemical. Chemical is a foreign entity, and it will elicit protective responses from organisms, cytochromes, different pathways to destroy those chemicals, to become resistant to those chemicals. RNA is very stealthy. A lot of the molecules that we use are actually present in the organism, are part of the organism's own regulatory systems. But nevertheless, we will see resistance. The way in which we address resistance is two ways. Number one, being stewards of the technology. We need to use integrated pest management strategies to ensure that we delay resistance as much as possible. And we are already partnering with multiple potential partners in figuring out how do we alternate RNA with other potential biologics, how do we use RNA in a sensible manner to delay the advent of resistance. But I think the most important tool that we have is the fact that we have the green box and that we can target a multitude of different genetic triggers in each one of those organisms. And so development of new actives against the same organism is not something that is as difficult as when you have chemicals. Because the reality is, within our bioinformatics capability and our high throughput capability, we're generating 100, 200, 900 in the case of botrytis, potential product candidates. And so when one product pathway becomes unavailable, you have multiple others that become candidates. Now, there are mechanisms of RNA resistance by themselves, right? There's mechanisms where an organism can become resistant to RNA. What we have seen historically is that the fitness penalty to RNA resistance seems to be too high for those organisms to survive. Ultimately, there's not going to be anything better than the responsibility of stewarding technologies. And we hope that by GreenLight, in many ways, blazing the trail and creating new pathways for discovery and development of new biological solutions, others will follow. Not necessarily with RNA, but with multiple other biological solutions that can be effective and safe.
How is there any example that has been shown in the presentation contain a mixture of RNAs or a hybrid RNA targeting multiple targets or different regions of the same target?
That's a good question. So in general, most of the triggers that you have seen are single triggers. Now, those are triggers that, as Mark talked about and as Ken talked about, are targeting more than one messenger RNA targets. So there are loci that can target the RNA triggers across a multitude of different species. Now, going from one RNA trigger to multiple RNA triggers is just a question of cost of goods. And so in cases where we decide that it's advantageous to have a multitude of double-stranded RNA species to target multiple regions of the gene or different genes, we can do that so long as the use rate allows it based on the cost of goods. I believe that's right. All of the triggers that we showed today are monospecies. Yes.
So we have a question online from Milan Oliver. So who are the current competitors, if any, that are working to solve the same agricultural problems with a similar technology?
So let's talk about competitors, and let's talk about collaborators or partners or people that are working on the same problems. Everybody's working on these problems. These are the biggest problems faced by the human species. We need to be able to feed 10 billion people with less land than what we're using today. This is not a small problem. This makes getting over a pandemic look like child's play. This is the biggest challenge of our generation. So everybody's working on this. Unfortunately, the majority of people who are working on this are still working on chemical solutions. Just think about it. We're going to be 10 billion people in the next 15 or 20 years. And that's how long it takes a new chemical to be developed. Order of magnitude. So by the time those solutions become available, it's going to be too late. But all of the major agricultural companies have their eye on the same issues. Resistance, insect resistance, fungal resistance, weed resistance, destruction of the fertility of our soil when we spray these chemicals that are extraordinarily harsh. Now, there are a multitude of other companies that are looking for alternative solutions. Microbial solutions, protein solutions, peptide solutions. The problem with those is it takes too long for those to be developed. And a lot of them require what we call baby treatment on the field. Baby treatment means you can't spray them from an aircraft. You can't put them on an irrigation system. You need to keep them refrigerated. And some of those are great solutions, and they have a lot of potential. The problem is farmers don't want to keep a big refrigerator in their farm to store an insecticide. They don't want to have to change how they spray. They don't want to change the intervals in which they spray. They want to know what the active is doing to the pest and to the crop. The problem with a lot of these microbial solutions, we don't actually know what they do. We have to keep them refrigerated. We can't spray them on a tractor, and we don't actually know what they do. And so that's a problem, because you have to be able to convince the consumer that what you're doing is safe, that it's not going to affect their health or the health of their children. So there's, unfortunately, a small number of firms that are pursuing highly defined biological solutions that are safe and cost-effective. What my hope is that by showing people that we can address this multi-billion-dollar market, many other companies will start and will get funded and will get traction in developing novel solutions. This is a market that is enormous. We're talking hundreds of billions of dollars that get spent in crop protection every year. The market is big enough for multiple competitors. We just need this entire ecosystem to grow up and to bring solutions that can be effective and that can actually restore a balance of equilibrium in the way in which we grow food. Yes.
John Roberts, Credit Suisse. It sounds like most of the Gen 2 and Gen 3 products are envisioned to be developed with partners. What do you see as the main challenges to developing partnerships and finding mutually beneficial arrangements with companies that do a lot of the same things you do downstream of manufacturing?
Yeah, that's a great question. I think the biggest challenge that we have historically faced is, will RNA be an approvable mode of action? And hopefully that will be significantly mitigated in the next few months. I think a lot of people are sort of waiting on the sidelines to see that EUP from the EPA, which is going to be kind of the blessing from the EPA that these products are safe, that they can be used in broad acreage. Unfortunately, a lot of the incumbents have franchises that compete with what we're doing. We were actually in France 2 weeks ago talking to a potential partner, and we found out in talking to this potential partner that the European Union had finally declined to extend the moratorium, am I saying that right, on neonicotinoids. So as many of you know, neonicotinoids were banned in the European Union, and then some exceptions were made to this ban so that farmers wouldn't completely freak out. And so a moratorium was put into this law that completely banned neonicotinoids, and that moratorium needed to be restored or referenda every year, extended every 12 months. And everybody expected that governments of the local countries, France in particular, would just go ahead and extend that moratorium. Well, it turns out the French government said, non plus, we're not extending that anymore. And French farmers freaked out, and they apparently descended. We arrived a day later, right? One day after this mess happened, apparently they just went to Paris with their tractors and blocked the entire city because they had already bought the seed for the entire season that contained neonicotinoids, and now they don't get to use it. And so what we're seeing is that, you know, a lot of the incumbents have franchises that they are trying to protect. They have shareholders that are demanding, you know, profits, which is the thing that shareholders tend to do. And so they're protecting these franchises, and it may not be the most easy thing for them to do, to adopt new technologies that, you know, compete with some of their existing portfolios. Now, the good news is that we're seeing systematic change across society and across regulators. We're beginning to see the health of our planet and the biodiversity of our ecosystems take front and center in conversations at very high levels. We're seeing the health of pollinators become a major concern to governments around the world. We're seeing chemical load on foods be a major concern to consumers and consumer groups. And so I believe that over the next 5 years, we're going to see a significant warming up, if you will, of the traditional incumbent agrochemical companies to biological solutions. We're beginning to see them sponsor frameworks for development of biological solutions. And so I believe it's only a matter of time. The biggest barrier for adoption is going to be regulatory processes. And that is something in which we are working extremely hard. We need to be able to show regulatory authorities around the world that RNA and other biological products are safe, that they can be an effective means to grow food more sustainably. And it is really up to us to carry the torch and demonstrate that. Mark, you want to add something?
Yeah, so if I can, I think when you look at the big companies who we're looking to partner with, what we're looking for is to trade our technology, our capabilities in RNA for boots on the ground. Okay, so we're looking for people who have territorial reach. Those tend to be the large manufacturers, and they are extraordinarily good at what they do. Synthesizing chemistry, discovering new molecules is what they do every single day of the week. But they don't discover highly functional biologics. Okay, we do. You've seen data today from Sean in the field, very high functioning biological materials. And most of the larger companies have recognized that that is not their skill set. They're going to insource that technology. Okay, and the discovery of high-functioning biologics. Andre referenced a load of companies who are working to come up with alternative technologies that are high-functioning biologics. There aren't many on the market right now. We're still in medium-functioning or low-functioning biologics. So they're insourcing that. So we have that. We can demonstrate that. We can demonstrate that not only are our high-functioning biologics high functioning. Sean showed lots of great data. We can show that they're highly specific, that we don't have to worry about any metabolites from any microorganisms that we put into the soil that we don't look for today. We have very clean technology. So we have a great technology swap to offer there in return for their development expertise, regulatory reach, right? There's 120 countries out there that you have to put a dossier through the door. So they have some of that reach, and we have a technology that they're not necessarily looking for themselves. And that's how the partnership builds, if that helps.
That's a much better answer. Take that answer. All right, let me thank you all for coming today. Thank you for your questions. Thank you for your attention. For those of you who are here in person, we're going to do a tour of our facilities, which should be a lot of fun. And so, and we also have some goodies that you can pick on your way out. So thank you all. Thank you for your attention. We look forward to seeing a number of you on Thursday. And as always, if you have any questions, don't hesitate to reach out to myself, to Susan, to Ingrid, to Nina, and so on. Thank you all.